region. Hi, I'm Juliana Strother, and today I'll be presenting on the cloning of the molecular nature of the Spemann-Mangold organizer, or specifically noggin, cordon, and follistatin. In 1924, Hilda Mangold and Hans Spemann conducted an experiment with xenopus, in which a piece of the dorsal lip of the blastopore of a donor was transplanted into a recipient in a region outside of the dorsal lip. They found that the dorsal lip of the donor blastopore was able to induce a secondary body axis at the site of transplantation in the recipient. They called the organizer region that was transplanted the Spemann-Mangold organizer, or Spemann organizer. They did not have the tools available to identify the molecular nature of the Spemann organizer at this time. Only in the 1990s, with new molecular biology tools available, did scientists begin to determine the molecular nature of the Spemann organizer. The first gene that was identified as part of the molecular nature of the Spemann organizer was noggin. Researchers had found that UV irradiation of the vegetal hemisphere of xenopus embryos resulted in only ventral structures forming in this embryo. They also found that injecting the RNA transcript of the Spemann organizer was able to rescue normal axis development in these ventralized embryos. This finding suggests that there is some gene or set of genes expressed in the Spemann organizer that can induce dorsalization and therefore is likely involved in axis formation during development. In 1992, Smith and Harland ran an assay in order to identify which gene or genes might be responsible for the dorsalization effect of the Spemann organizer. They collected mRNA from xenopus embryos that were hyperdorsalized during the blastula stages through lithium chloride treatment. They knew that dorsalization gene could be overexpressed in these embryos, and therefore the mRNA present would align with the dorsalization genes being expressed. They then converted this mRNA into a library of cDNA, or plasmid DNA, with fragments of different sizes, and injected the cDNA into the irradiated embryos. The pools of plasma DNA that were able to induce dorsalization in the irradiated embryos were selected. A new library was generated, and this process was continued until the clone with the smallest region of DNA that conferred this desired effect was identified. The gene that this clone expressed was sequenced and named noggin. Two follow-up experiments supported noggin's role as an inducer of dorsalization in the Spemann organizer by showing that noggin mRNA injected into ventralized embryos can induce dorsalization, and that noggin is expressed in the Spemann organizer regions at the correct times of development that would align with its organizing activity, based on in situ hybridization experiments. Therefore, noggin was shown to be an inducer of dorsalization and part of the molecular nature of the Spemann organizer. The first indication that another dorsalization inducer is present in the Spemann organizer is that injection of homeobox gene called goosecoid, which is specific to the Spemann organizer, leads to the induction of a secondary axis when injected into the ventral side of an embryo. As a transcription factor, goosecoid causes the cell to express genes downstream of it, and noggin is not one of these genes, suggesting another dorsalization inducer is present in the Spemann organizer. The experimental design for isolating this dorsalization inducer, cordon, was very similar to what was used to isolate noggin, except with the additional confirmation step of whole mount in situ hybridization, which was used to identify the spatial and temporal dynamics of candidate gene activation by organizer-specific homeobox products resulting from goosecoid injection into embryos. The only gene that was shown to be activated by goosecoid injection and exclusively expressed in cells with Spemann organizer activity was cordon. Cordon was also shown to be able to induce a secondary axis when injected into the ventral side of the embryo, and therefore was shown to be an inducer of dorsalization and part of the molecular nature of the Spemann organizer. The two central properties of the Spemann organizer are its ability to induce dorsalization, like noggin and cordon were shown to do, and its ability to induce neuralization. Inducing neuralization is caused by BMP inhibition, as BMP converts ectoderm to epidermal tissue rather than letting it default to neural tissue. Noggin and cordon were shown in 1993 and 1996, respectively, to also serve as BMP antagonists. The final major component of the molecular nature of the Spemann organizer is follistatin, which was initially identified as a neuralization factor in 1994, before being shown to have a dorsalization effect in 1995. Previous studies had shown that dominant negative activin receptors resulted in ectoderm becoming neural tissue, which led Hamadi, Brinvenlo, and Melson to conclude that activin antagonists contribute to neural tissue differentiation. They already knew that follistatin is able to bind and inactivate activin, and ran a series of experiments to prove that follistatin is a neural inducer expressed in Spemann organizer. First, they identified the homolog of follistatin that was present in xenophos embryos and showed that it is a secreted protein in the embryo. Reverse transcriptase PCR showed that follistatin is encoded maternally in xenophos, and whole mount in situ hybridization showed that follistatin RNA is expressed in the embryo, beginning with gastrulation, is initially localized to Spemann organizer cells, consistent with neural induction activity. Injection of follistatin RNA was also shown to directly induce neural differentiation. All of these findings supported follistatin as a neural inducer and part of the Spemann organizer.